my YouTube channel. Today, the topic that we are going to discuss today is PCR. This is a cell biology technique. So let's move forward. So PCR was invented by Carey Millis in 1983, who also got a Nobel Prize for the same. He was a biochemist, and PCR stands for polymerase chain reaction. It provides a powerful solution, which is it makes millions of copies of a specific DNA region from a minuscule starting sample, which means from a very low amount of sample, it can make millions of copies of a specific DNA. This process relies on a thermal cycler to precisely control a series of repeated temperature changes, exponentially amplifying the target sequence. In PCR, we majorly have three steps, which are the denaturation, which is done at 95 degrees Celsius. In denaturation, the double-stranded DNA template is heated to separate it into two single strands. So here is our template DNA, which is double-stranded. When we treat it at 95 degrees Celsius, it becomes single stranded. Then the next is annealing. In the annealing is done at 55 degrees Celsius. In annealing, we require two types of primers. One is the forward primer and one is the reverse primer. The temperature is lowered to allow short DNA primers, which are of length 18 to 22 nucleotides, to bind, which also means anneal, to their complementary sequences on the single-stranded templates. So the primers anneal themselves on the single-stranded templates. After the annealing, we have extension, which is done at which is done at 72 degrees Celsius. So from 55 degrees Celsius, the temperature is increased to 72 degrees Celsius. And at this temperature, the polymerase, which is the tag polymerase used here, it binds to the single stranded DNA with the help of the free 3' end OH of the primer and then extends it. The temperature is raised and the enzyme tag polymerase synthesizes new DNA strands extending from the primers from their 3' OH end. So the number of DNA molecules is doubled. This cycle is repeated 25 to 35 times, which results in the exponential growth. So from denaturation, annealing and extension, this is one complete cycle of a PCR, and this cycle is repeated 25 to 35 times. So moving forward, so the molecular toolkit for PCR. A successful PCR reaction is a finely tuned interplay of several critical components, important components, each with a specific role in the amplification process. So there are various components that we require which play a specific role in the amplification process. The first one is our DNA template, the sample, which is the sample containing the target sequence to be amplified. Then there are primers, which are short single-stranded DNA fragments, which are also known as oligonucleotides, which is complementary to the 3' end of the target sequence. They provide the starting point for DNA synthesis. So the primers are around 18 to 22 base pairs of length, nucleotides of length, and it has a free 3' OH end and it binds to the DNA template which is single stranded. Then there is our tag polymerase. The tag polymerase is a thermostable DNA polymerase which is originally taken from the E. coli, a Thermus aquaticus, that can withstand the high temperatures of the denaturation step. It enzymatically assembles the new DNA strands. Then there are our dNTPs, which are the deoxynucleoside triphosphates. They are of four types: A, T, C, and G. The A, T, C, and G, adenosine, cytosine, guanine, and thymine, are the building blocks used by the polymerase to construct the new DNA strands. These are our bases. And then there is buffer solution and Mg2+, which creates the optimal chemical environment, which is the pH and ionic strength for the best polymerase activity. The magnesium 2 plus ions act as an essential cofactor for polymerases. Moving on to the next slide. So we have a specific challenge, the specificity challenge during PCR, which is ensuring we copy the right target. The specificity of PCR is determined almost entirely by the primers. Poor primer designs lead to non-specific amplification or complete reaction failure. Good designs ensures the reaction is precise and efficient and there is no unnecessary binding. So key principles of effective primer design are the length. The length of the primer, as I already said, should be between 18 to 22 or 24 base pairs, which is the standard. Too short and the specificity is lost, and too long and binding efficiency can decrease. So if the primer is too short, then they won't be specific. There will be non-specific. If they are too short, there will be non-specific binding, and if they are too long, then the binding efficiency can decrease, which means the primer can't bind efficiently. Then there is melting temperature, which is also known as Tm. The optimal Tm is between 50 to 70 degrees Celsius. The Tm of the forward and reverse primers in a pair should be compatible. The max difference between the Tm value of the difference of the forward and reverse primer should be 3 to 5 degrees Celsius. The max Tm value between the forward and reverse primers in a pair should be between 3 to 5 degrees Celsius. Then there is GC content. The GC content in the primers should be between 50 to 60%. This ensures appropriate hybridization stability. The common pitfalls which we should avoid is that we should avoid the formation of secondary structure, like primers must be designed to avoid forming hairpins, which means folding onto themselves. So the primer sequence shouldn't have complementary sequences at the ends, which can result in binding of the either ends. Or there shouldn't be dimer formation, which means binding to each other, especially at the 3' end, as this can block amplification. So if the two primers, the forward and reverse primers, are complementary to each other, then they can form a primer dimer, which means they can anneal to each other, and hence the polymerase won't bind and there will be no amplification of DNA. Moving on to next. So to make the for the making amplified DNA visible after PCR, the amplified DNA fragment must be separated and visualized to confirm the reaction success and determine the product size. Agarose gel electrophoresis is the standard method to achieve this. The principle of agarose gel electrophoresis is that first of all we have the matrix, and agarose gel is a gel which has many pores, and these pores form a matrix like structure which has many gaps in between. Then an electric field is applied when the gel is submerged in a buffer, and an electric field is applied on the gel. When the electric field is applied, since the DNA is negatively charged, it migrates through the gel towards the positive electrode, and then there is separation. The smaller DNA fragments move through the pores more easily and travel further than larger fragments, achieving separation by size. So if we load small fragments of if we load a DNA sample, then the DNA will be separated based on their size, as the smaller fragments will move faster towards the positive end and the larger fragments will be left behind. Then there is visualization. A fluorescent dye, which is ETBR, intercalates into the DNA and fluoresces under UV light, making the DNA bands visible. A DNA ladder with fragments of known sizes is run alongside for comparison. So in the first line here it is shown that a DNA ladder is run alongside for comparison of the fragments, so that we can judge what is the length of our DNA fragment. So this is the setup. Here we have the gel tray in which the agarose gel is kept. then a buffer is added and electric current is applied from negative to positive. Then the samples are loaded, and this is known as loading. We load the DNA sample here, and then after applying the current, the charge, the DNA starts to migrate. We also add the ladder as shown here, which is of 1000 base pairs, and the DNA starts to migrate based on their size and separate towards the DNA is, as I already told, is negatively charged, so it begins to migrate towards the positive charge. And on the result, when we see, we visualize it under UV light, we can see the bands are formed, and we can determine if our amplification is done or not. So moving on to next, while conventional PCR followed by gel electrophoresis answers a critical question, is the target DNA present? It falls short in many applications like for diagnostics, gene expression analysis and viral load monitoring. A different question is paramount: how much target DNA is in the sample? So the limitations of the conventional PCR for quantification are the through conventional PCR we can determine if the DNA is present or absent, but we can't quantify it. So there is endpoint analysis. Gel analysis only shows the final product after all cycles are complete. The plateau, which is the plateau stage, which is not proportional to the initial amount of template. It is imprecise, which provides only a rough estimate of quantity. We cannot determine the exact quantity of the sample DNA. And then PCR, and the conventional PCR is also time consuming, which requires a post PCR processing step, which is the gel run. So what is the need? The need is to quantify. The need a method that can measure the amount of DNA as it is being amplified. So from the starting only we can amplify, that is the need. So the breakthrough for this was watching DNA amplify in real time with qPCR. qPCR stands for quantitative polymerase chain reaction. The quantitative PCR revolutionized DNA analysis by enabling the monitoring and measurement of DNA amplification during each cycle of the reaction in real time. This is achieved by incorporating fluorescent molecules into the reaction. So first of all, there is a DNA binding dye, for example cyber green, which is a fluorescent dye. It fluoresces when bound to any double-stranded DNA. So whenever the cyber green binds to the double-stranded DNA, it gives fluorescence color in real time. So when there is a PCR running, we get the real-time quantification of how much DNA is amplified in each cycle. And then there is sequence specific probe, which is uh, a single stranded oligonucleotide, and it has a reporter at its one end and quencher at its other end. It binds to the single stranded DNA, and when a reporter and quencher binds to a specific sequence, when the polymerase starts making a new DNA strand, it cleaves the probes, separating the dyes and allowing the reporter to fluoresce. So in the presence of the quencher, the reporter doesn't show any fluorescence. But when the DNA polymerase, while making a new strand, removes the reporter from the quencher, it shows fluorescence because it is now free. So here is our fluorescence intensity, which is also known as RFU, and here is in the y-axis, and in the x-axis we have our cycle number. So when there is this is our fluorescent threshold, and the CQ stands for quantification cycle. The quantification cycle value is the cycle number at which the fluorescence crosses a set threshold. A lower CQ value indicates a higher initial amount of target DNA. So if we have a low starting quantity, the CQ value shows that we have a higher initial amount of target DNA. And if there is high starting quantity, then there will be higher CQ. So here there is high CQ, here there is low CQ. Moving on to next. So qPCR in action. The development of a reverse transcription qPCR assay for Geta virus highlights the technique's diagnostic power. Geta virus poses a serious threat to livestock, requiring a rapid, sensitive, and specific method for epidemiological investigation. So here is a case study in virus epidemiology where we are using qPCR, which is quantitative PCR, for detection. So the challenge is to establish a reliable method for detecting and quantifying Geta virus in serum and mosquito specimens. So the solution is, as earlier discussed, and cyber green 1 RT-PCR real time reverse transcription qPCR assay targeting the highly conserved NSP1 gene. So there is a gene which is highly conserved in the Geta virus of Gateway. So there is high sensitivity. We can see minimum detection limit of 7.73 plaque forming units per microliter. Then there is superior performance. The RT-qPCR was 100-fold more sensitive than conventional RT-PCR. Then there is excellent linearity. A strong linear relationship was found between viral load and CQ value, which is R square is equals to 0.998, enabling accurate quantification. The diagnostic value receiving operating characteristic curve analysis confirmed high diagnostic value AUC equals to 0.956 with an optimal CQ cutoff of 37.59. Moving on to the next, the real world challenge when samples fight back. In many critical applications, especially in forensics and clinical diagnostics, the DNA samples are not pure and there are lots of background uh, data also, which background noise also. Since our DNA is not pure, they are derived from complex matrices like soil, blood, or tissue, which contains substances that can interfere with or completely block the PCR process. These are known as PCR inhibitors. The impact of inhibition is that it reduces amplification efficiency and leads to underestimation of DNA quantity, higher CQ in qPCR. And the impact of inhibition is also that there is complete amplification failure, which results in false negative results. Then there are common sources of inhibitors which are in forensic samples, which is humic acid from soil, indigo dyes from denim, hemoglobin from blood. So these all inhibit the PCR process. And then there are in clinical samples the inhibitors in like hemoglobin, immunoglobulin G, and anticoagulants like EDTA, heparin in blood, bile salts in feces. Then there are some PCR inhibitors in sample processing like phenol from DNA extraction and cellulose from filter papers. So the mechanism of inhibition. Now we will discuss about the mechanism of inhibition and how it inhibits. So different inhibitors disrupt PCR through distinct molecular mechanisms. Understanding these mechanisms is key to developing effective countermeasures. So the first inhibitor is our humic acid, which is from the soil. So the mechanism of action is it does polymerase interference. That means it directly lowers the activity of DNA polymerase, likely in the early PCR cycles. Then there is fluorescence quenching. The humic acid binds to double-stranded DNA dyes, example silver green, evergreen, and causing static quenching and reducing the fluorescent signal. Then there is primary effect, which is the decreased amplification efficiency and false negatives or under quantification in quantitative PCR. Then the next inhibitor, commonly known inhibitor, is hemoglobin, which is from blood. It also causes polymerase interference, which lowers DNA polymerase activity throughout the PCR, just like in the humic acid. It also does fluorescence quenching, which means it binds to and quenches double-stranded DNA binding dyes and reference dyes like ROS. Then there is decreased amplification, and its primary effect is decreased amplification efficiency, skewed qPCR results due to reference dye quenching. Then there is immunoglobulin G, IgG, which is also from blood. It does what? It does template masking, which is preferentially binds to single-stranded genomic DNA. Single-stranded DNA. It binds to the single-stranded DNA IgG, thus physically hindering primary annealing and initiation of amplification. So the IgG binds to the single-stranded DNA and it prevents the binding or annealing of the primers, and hence initiation of amplification is only delayed or stopped. So the primary effect is delayed amplification without affecting the slope of the amplification curve. So the breakthrough, a toolkit for overcoming inhibition. So a combination of strategic sample preparation, optimized reaction chemistry and enzyme technology can effectively neutralize the impact of PCR inhibitors. So the first, for overcoming inhibition, we can do what is sample dilution. Sample dilution is simple but risks diluting scarce target DNA below the detection limit. Then there is enhanced purification. Methods like using Chelex resin or magnetic beads can remove inhibitors but may lead to significant DNA loss. Recovery can be as low as 10%. Then there are inherent inhibitor tolerant DNA polymerase. The most polymerases, the most direct solution. Screening and selecting robust polymerases significantly improves performance with challenging samples. Then there are PCR additive facilitators. The PCR additive facilitators are such as BSA bovine serum albumin binds to inhibitors like humic acid only and betaine trehalose, which helps destabilize the DNA secondary structures. Then there is detection strategy. For samples with humic acid or hemoglobin, we can detect it using hydrolysis probes, example TACMET. Instead of double-stranded DNA binding, dyes avoids the problem of fluorescence quenching. The next one here. So the next frontier, pushing the limits of sensitivity and speed. As scientific questions become more complex, PCR technology continues to evolve. Next generation methods are solving the challenges of absolute quantification, extreme sensitivity, and point of care analysis. So then there is to push the limits of sensitivity and speed and go from conventional to more modern versions of PCR, there is the digital PCR, which is also known as dPCR. The challenge, precisely quantifying rare targets or low copy DNA where traditional qPCR can be inaccurate. The breakthrough, the reaction is partitioned into thousands of individual micro reactions. After amplification, the number of positive partitions is counted. The advantage, provides absolute quantification without a standard curve and shows remarkable resistance to PCR inhibitors. Then there is microfluidic PCR, which is also a lab on a chip type. The challenge is the need for rapid on-site DNA analysis outside of a traditional lab setting. Breakthrough miniaturized systems that perform PCR in microscale channels and chambers. The advantage is that it requires nanoliter scale volumes, very low amount of volume, significantly reducing reaction times and enabling portable automated systems for field deployment. So this is our practical guide to PCR troubleshooting. Like, while powerful PCR, we know that PCR can is a very powerful molecular technique, but it can often fail. So this is a systematic approach to troubleshooting, or which can quickly identify and resolve common issues. So the problem is, first of problem number one is that often we have no amplification or low yields, whose common causes can be poor quality or quantity of DNA template, suboptimal annealing temperature or MgCl2 concentration, insufficient enzymes or dNTPs. The recommended solution for no amplification or low yield are repurify or concentrate DNA template, optimize annealing temperature, typically 3 to 5 degrees Celsius below primer melting point, titrate MgCl2 concentration. Then there is another problem which exists, which is the non-specific products or extra bands which we often get. So the common causes for this are annealing temperature is too low. There is non-specific primer banding during reaction setup. So the recommended solutions are increase the annealing temperature, use a hot start polymerase that is only activated at high temperatures, or to redesign primers for a better specificity. The next problem is primer dimer formation. So the common causes are high primer concentration or complementarity between primer sequences, especially at the 3' ends. The recommended solutions are reduce the primer concentration, increase annealing temperature, redesign primers to minimize self complementarity. Then another problem which exists and often is seen in labs is the formation of smeared bands. For that, the common causes are degraded template DNA, extension time too long, contamination with amplifiable DNA, contaminants specific to the primers, and the recommended solutions for this is use high quality intact DNA, optimize extension time. Rule of thumb is 1000 bases per minute for tag polymerase. If contamination is suspected, switch to a new set of primers. So the next is the evolution of the genetic resolution. The journey of PCR is a story of escalating challenges met with ingenious breakthroughs. What began as a simple method to simply make DNA visible has evolved into a sophisticated suite of tools capable of precise quantification, high throughput analysis, and robust performance, even with the most difficult real world samples. Each innovation has brought the molecular world into sharper focus. So, the trajectory of resolution I have shown here that the fundamental challenge in the starting that we faced was to see the unseen, like we can't visualize the DNA with our naked eyes. For that, we used to detect its presence or absence, we used the conventional PCR. Then we wanted to check how much quantity is present. For that, to detect the precise quantity of the DNA, we used qPCR, quantitative PCR, and then we have upgraded to absolute count, which is that we can see how many copies per microliter are made in dPCR, direct PCR, and in complex and inhibited samples. So right from seeing the unseen to the absolute count, we have covered a long way, and more discoveries are yet to made in molecular bio field. And if you enjoyed this video, feel free to like, share and subscribe. Thank you.